Okay, so that's a bit about the background. I'm going to try now to get involved a little bit in some of the techniques, kind of the big question, right? So where do I begin? I'm somebody who works on protein X. I want to know, if microRNAs are important in my system. So usually, I try to group this into a forward genetic approach versus a reverse genetic approach. I guess you can do this with any system. Scheme 1 is kind of laid out as, I guess, a forward genetic. So essentially, you did a big profiling experiment. You can do this by a microRNA microarray. You can do this by small RNA sequencing. And there's many other techniques as well. And then you do this maybe normal versus tumor, or some disease state versus normal, or you treated cells with a drug, and you have a vehicle control, whatever it may be. You do the profiling, you find a bunch of microRNAs that change upon your treatment, and then you have to do the hard task of identifying and validating your candidates, and then move on to some functional experiments. The other approach could be the reverse genetics, I guess, where it's more, maybe it's bioinformatic, maybe it's, you already have a phenotype, right? So you're now asking the question, I have one particular gene, I know it promotes something like aberrant cell growth, and now I want to know which microRNAs are interacting with my gene, or maybe you already know a microRNA does something and you want to know what genes are regulated by that microRNA. That's sort of a reverse genetics approach. You kind of already know your phenotype-ish, and now you're just trying to ask, what's the upstream regulators? There, it could be a little bit easier, but I mean, you still have to then go and screen for the expression of your microRNAs to see if they're even present in your cell, and if they inversely correlate with the mRNA of your target gene. And then eventually you'll still have to go back to Scheme 1. I mean, these are not separate approaches. It's just how you enter into the system. If you start with Scheme 2, you eventually will have to do Scheme 1 to some extent, and vice versa. And then overall, I think what's really important, if somebody's not used to working with RNA, you do have to be a little bit more careful. I mean, RNA has some of its own intricacies, and it's really important to know a little bit about RNA biochemistry, and you still have to do a lot of validation experiments. So that's kind of the roadmap, depending on which angle you're coming from.
To view the full video of this and all of our other webinars for bioscientists at the bench, please visit bitesizebio.com/webinars.